Welcome back to Botany class. In the previous class, we concluded with steps involved in recombinant DNA technology. Today, let's begin from tools of genetic engineering. Tools for genetic engineering. In order to generate recombinant DNA, there are some tools are involved. The basic tools are enzymes, vectors and host organism. What are the tools involved? There are certain tools involved for genetic engineering technology. Previous class we discussed about the topic genetic engineering. Recombinant DNA technology is very much used in this. For making recombinant DNA, there are some tools involved. What are the tools? Enzymes, vectors and host organisms. Enzymes, vectors or host organisms used as the basic tools. The enzymes used for this tool is restriction endonucleases, ligase, alkaline phosphatase. There are three different enzymes are involved, namely restriction endonucleases, DNA ligase and alkaline phosphatase. These three enzymes are necessary for this genetic engineering technology. Today, let's see that restriction endonucleases, the action of restriction endonucleases, ligase and alkaline phosphatase. Restriction endonucleases. Some bacteria, there is a common bacteria, you know, Escherichia coli. Commonly it is called as E. coli. So, in this bacteria, Escherichia coli, there are some enzymes. There are two different kinds of enzymes observed in Escherichia coli. The one enzyme which adds methyl group CH3. So, some enzymes, one enzyme, there are two enzymes discovered in the year 1963 in E. coli bacteria. E. coli bacteria which is infected by bacteriophage. So, this bacteriophage, in bacteria certain enzymes are secreted. So, these types of enzymes, restriction enzymes are restricted the growth of foreign invaders, that is disease causing microbes, bacteriophage. What is bacteriophage? The virus which attack bacteria. Here this E. coli bacteria which attacked by bacteriophage. So, for defense mechanism, this enzymes are secreted in bacteria's body. So, this enzyme can be isolated. So, here it was found two enzymes, that is one which adds the methyl group CH3. So, CH, this is one enzyme which adds the methyl group, while the other enzyme which cuts it used for cutting the DNA sequences. So, the second type, later type, it's called as restriction endonucleases because it cuts the DNA sequence. It's named as restriction endonucleases. So, this restriction endonucleases depends upon its mode of action or its function. Its mode of action, there are two different types of restriction endonucleases, namely exonucleases and endonucleases. Exonucleases, endonucleases, that is restriction endonucleases are two types, depend upon its mode of action. Exo, as you know the meaning, exo and endo. So, exo endonuclease and endonuclease. So, this exonucleases, so as it indicates exo, outside, which cuts the DNA at one end, at the end of any one of the DNA sequence, at the end it cuts, exonucleases. Example, ball 1, exonucleases 3 are the examples of this exonucleases. Endonucleases within the DNA sequence, internally, inside, in the, here it inside, in the phosphor diester bond, inside it cuts, endo means inside. So, within the nucleotide sequences it cuts. So, there are two restriction enzymes. One is cutting outside, one sequences, any one of the sequence which is present outside, that is exonucleases. Endonucleases which cuts the DNA sequence internally. So, based on its mode, it is classified into two different types, that is exonucleases and endonucleases. Then these enzymes, these restriction endonucleases are named as molecular scissors. It's named as molecular scissors. Due to this action, this restriction endonucleases are very important. So, this scissors, you know the use of scissors which used to cut, these are named as molecular scissors. These enzymes which used to cut the DNA sequences. So, already previous class we learned that it's not an easy job to cut and paste it. But here these all enzymes under this genetic engineering technology, there is an alteration, that means manipulation of DNA sequences. All the DNA sequences can be altered or manipulated by the use of these all enzymes. So, restriction endonucleases plays very important role to cut the DNA sequence. And another enzyme is there, ligase. Ligase enzyme used to join the sugar and phosphate group. So, in the nucleotide which consists of a nitrogenous base, sugar, this is the sugar, here is a nitrogenous base, that is adenine, guanine, thymine, cytosine, and there is a phosphate group. So, this is a nucleotide, this is a base, nitrogenous base attached with sugar and phosphate group. So, in this nucleotide sequence, these restriction enzymes are used for cutting. It cuts and it pastes at any direction or a particular site, and it is very important, molecular scissors. Restriction endonucleases, actually it is present in most of the bacteria, almost all bacteria, restriction endonucleases are present. Why it is present? It uses them for defense mechanism to escape from the foreign invaders. If any disease causing organisms are enter into its body, it cuts it, the sequences, it cuts the nucleotide sequence and it uses for the bacteria to escape from the disease causing agents. That is the function of restriction endonucleases. Now, this restriction endonucleases are involved in this technology, genetic engineering technology, to alter the DNA sequences. So, for altering this, vectors are important. Then this enzyme, what is the role of this restriction endonucleases? It recognizes a specific site. It cuts in a specific site. Because in the nucleotide, you know that it cuts in, it joins the complementary pair in the 3-5- direction. Adenine, guanine, thymine, cytosine. The complementary pair is here adenine combines with thymine, guanine with cytosine, thymine with adenine, guanine with cytosine. So, 5-3-. So, this is the complementary pair. So, there is a rule. Adenine combines with thymine, guanine combines with cytosine. So, this is a rule. So, it recognizes a particular site in the DNA sequences and it cuts. So, in a particular sequence, the restriction enzyme selected the area called as restriction site. Restriction site, or it is called as recognition site. In the recognition site, it cuts in a particular sequence, specific area. So, there are 3 different types of the restriction enzymes are classified into 3 different types: type 1, 2 and 3. Here in recombinant DNA technology, that is rDNA technology, this type 2 restriction enzymes are involved. So, one of the example is Hint2. This is the restriction enzyme used to cleave in the base pairs, particularly in this base pairs. It cuts in a specific sequences there 4 to 8. It cuts in between 4 to 8 base pairs commonly. So, this mainly this Hint2, this Hint2 which used to cut the 6 base pair. 6 base pair Hint2 cuts. So, there are 3 different types according to this classification restriction enzymes. There are 3 different types: type 1, type 2 and type 3. So, based on this, type 2 is used to cut the DNA sequence, that is it is used for restriction endonucleases, for cleavage. What is the role of this? It cleaves the DNA sequence. You know that when you are making a recombinant DNA, the plasmid or the vector can be cut open and a foreign DNA can be inserted. So, cutting and insert the DNA sequence, for making a cut, the molecular scissors are used as restriction endonucleases. By the use of this enzyme, the plasmid as well as the desirable gene from the fragments of desirable gene selected from isolated organism can be cut open. So, 2 cells can be cut open, plasmid as well as the foreign DNA sequences, and it is inserted inside the vector by means of restriction endonucleases. And usually this restriction endonucleases are palindromic in nature. Palindromic. What's this palindromic? So, there is a phrase Malayalam. See, in this phrase, see in this direction or this direction. See, when you write from here M A L A Y A L A M. So, both the sides, this direction and this direction, it read as the same. So, this is called as palindromic. So, in the recognition site of this DNA sequence, it is known as palindromic. For example, there is an example for this palindromic, that is there is a sequence, DNA sequence, you remember it in the form of you just remember cat, tat, at, odd, G. So from 5 dash 3 dash direction. So this is one complementary pair, is the complementary pair of this is, so here 3 dash 5 dash, what's the complementary pair? C always combines with G, A with T, here T, A, T, A, T, T, A, T, T, A, T, T, A, C. Say this, C, A, T, T, A, T, A, T, A, T and G. So this direction and this direction, both the direction it read the same similar sequence. So this sequence is called as palindromic. Palindromic repeats. So example, sometimes 2 mark question or 3 mark question they may ask palindromic repeats. So simply you remember, just for your remembrance, you read it as cat, tat, at, odd, G. So complementary pair you can write it. So this is example for palindromic sequence. So this restriction endonucleases cut in a specific sequences. And the name of the restriction enzyme, there are number of restriction enzymes. Now it is there are 900 restriction enzymes are isolated from 230 different bacteria. So this restriction enzymes are named as in a specific manner. So naming of this restriction enzyme, one of the common restriction enzyme is Escherichia coli, ECO-R1. So there is a naming, there is a specific manner to name this, is planned and named. So first letter, capital E, this first letter is represent, because this ECO-R1 isolated from Escherichia coli. So this named as first letter derived from genus name of Escherichia coli, E, and followed by these 2 letters species name, coli, E. coli, Escherichia, first letter genus name, followed by 2 letters represents or indicates the species name, and this R is the strain. Here in this strain is RY13 is the strain. So from there the first letter R, and the Roman numeral, it ends with a Roman numeral, which is the order of discovery. So this was the first restriction enzyme which was found. So it is Roman letter 1. So remember it, the naming of this restriction enzymes. How to name this? First, the bacteria, almost all this are obtained from bacteria, because bacteria which secrete number of enzymes, restriction enzymes. So from there the genus name and species name, Escherichia coli. So Escherichia is the genus name, coli is the species name, binomials. So first letter of this enzyme is named from the genus, followed by 2 letters, next 2 letters from the species name, and the strain R. Here RY13 is the strain. So that first letter is R, and finally it ends with Roman numeral 1, 2, 3. So based on this, the order of discovery. So this was the first enzyme to be discovered. So it's named as 1. The name of this enzyme is ECO-R1. Now there are some examples are present here. Look at this tablet column. the restriction enzymes. There are different types of restriction enzymes, namely ALU1, BAM, H1, ECOR1, HAE3, HINT3, HINT2, HINT3, so many enzymes are there. So, this is the tablet column which shows, it is given in your book, which shows that the restriction enzyme, name of the restriction enzyme and microbial source. What is the microbial source and what is the recognition site of it and what are the fragments. From this tablet column you may expect some one-word questions. So, you have to prepare this, you have to learn the types of restriction enzyme. First you learn these types and what are the microbial source. For example, ALU1, the source, the microorganism is Orthobacter luteus. From Orthobacter luteus, ALU1 is derived, and here is the complementary recognizes the sequences A, G and CT. So, when it cuts, there are two different ends are there. They are called as, according to the cleavage nature, the recognition site which cleaves either at the end or in the center. Based on this cleavage site, it can be divided into two different types, either blunt end or sticky end. The blunt end or the sticky end. So, two different ends are there. Blunt end which cuts in the center of the sequences, sticky end in the surface. So, the same enzyme cuts either in the center or in the surface. Depend upon it, if it cuts the center, it is called blunt end or flush end, or sticky end or cohesive end. So, based on this, here the complementary sequence. The complementary sequence of this is here A, 5' 3' direction, AG and CT. So, the complementary strand is adenine combines with thymine, here C, G, A. So, it cuts here. Here is the recognition site sequence. Recognition sequence is this area. So, A, G, C, T and T, C and G, A. So, it cuts in the center. So, it comes under blunt end. Then, BAM H1, Bacillus amyloliquefaciens. The name of the microbe or the source of microbe is Bacillus amyloliquefaciens. Here the complementary strand is C, C, T, A. So, it cuts in the sticky ends. Here this is the place it cuts. So, it cuts in the sticky end. In the surface it cuts. Then ECOR, already you learned this, Escherichia coli, and the complementary strand is C, T, T, A, A. It's also sticky end. And here C, C, G, G. Here T, T, C, G is the complementary pair. Then this source of this HAE3 is Haemophilus aegyptus. HINT3 Haemophilus influenza. So, this is the blunt end, this is the sticky end. The recognition fragment is here sticky end, here blunt end. Hope that you understand it. So, name all this. First you learn the name of the enzyme and learn the source of the microbes, source of the bacteria. This is the bacterial name, genus name and the species name, and which is the recognition site and what type of cut it. Based on this, what classification, either blunt end, blunt, or it comes under sticky end. So, blunt end or sticky end, it cuts either in the center, blunt end, or in the surface, that is sticky ends. So, restriction endonuclease, very important question, restriction endonucleases. There are three enzymes involved. First one is restriction endonucleases. There are some classification based on this and different types are used for this. Second, this is used for cleave the DNA sequence, to cleave, to cut the fragments, DNA fragments. After cleavage, it has to join. So, first there is a cleavage. In the rDNA technology, recombinant DNA technology, the plasmid as well as the desirable gene selected from isolated DNA can be cut open by use of this restriction endonucleases in the recognition site. Now, ligase, what is the use of ligase enzyme? This, you know that ligase enzyme which joins the sugar and phosphate group. Look at this, here the sugar and phosphate group, this in PO4 and OH. So, it joins this region in the diester phosphate group with the presence of ATP. So, there are some cofactors involved in this joining, either it is ATP or NAD+. If so, it joins the DNA sequences. So, it joins in the diphosphodiester bond. The ligase enzyme which joins sugar and phosphate in the double-stranded DNA. See, this is the double-stranded DNA. In the double-stranded DNA, it cuts internally with the help of this ATP or NAD+. Mainly this ATP, adenosine triphosphate, is involved and it joins in the phosphodiester groups. That is the role of ligase enzyme. Then another enzyme is there, that is alkaline phosphatase. Alkaline phosphatase, this is used as a modifier enzyme, enzyme modifier. Either it adds or remove the sequences from the 5-phosphatase terminals. In the 5-phosphatase terminals, either it adds or it removes. That is the role of alkaline phosphatase. Look at this. Here is the ligase enzyme. This ligase enzyme which joins this phosphate PO4 and OH. So, ligase enzyme PO4 and OH, it joins. But if the sequence is the correct sequence, it joins. But here there is no reaction because here POH, complementary pair. Here it is not like this, OH, here OH. So, there is no reaction in this. But this is the enzyme. So, here it joins. Here OH, POH. So, alkaline phosphatase. So, here this is the ligase enzyme, action of ligase enzyme. Here is the alkaline phosphatase enzyme. So, alkaline phosphatase enzyme, what is the role? It is the modifier enzyme. So, either it adds or removes in the 5-phosphate terminals. So, this point you keep it in your mind, this ligase and this alkaline phosphatase. So, restriction enzyme, prepare it for 5-mark question, ligase 2-mark question and alkaline phosphatase 3-mark question. So, 3 enzymes are involved as a tool. Apart from this, another major component is used for recombinant DNA technology. What is that? That is vector. So, these vectors. So, first enzymes. You learned there are 3 important enzymes, restriction endonucleases, ligase and alkaline phosphatase. Its role. First question you learned this. Second question, the another major component involved in this rDNA technology or recombinant DNA technology or genetic engineering is vectors. What are vectors? These are nothing but a plasmid. What's this plasmid? The plasmid, you know, in the bacteria, in the bacterial cell, in addition with this genome, there is a nuclear material, nucleoid, there is a extracircular DNA which is called as plasmid. So this plasmid is present in bacteria as an extracircular DNA, is very much used as a vector for cloning this DNA, foreign DNA. There is a vector need. So these vectors are otherwise called cloning vehicle. Cloning vehicle. Vectors are named as cloning vehicle or cloning DNA. So DNA fragments, what's the function of this? It can carry the DNA fragment that is selected, that is the desired gene, which is isolated from a particular DNA sequence. The host, and it has to introduce it into the host organism. So at first the plasmid can be isolated from the bacteria, and this plasmid can be altered by means of restriction endonucleases, cut opens, and the foreign DNA can be inserted by means of ligase enzyme. It can be pasted. Again it is introduced into host organism to multiply the copies. So what's the feature of this vectors? The vectors can be replicated. It should be replicated. It produces number of copies along with this inserted DNA molecule. That is the property of vector. One of the properties, what are the properties of vectors? It can be automatically, it is self-replicating. Plasmid can be self, or vectors can be self-replicated. And what are the properties of this? First thing, it can be self-replicated and it can be carried the foreign DNA along with replication. Along with the foreign DNA, it can be replicated. And what are the properties? It should be very small and the molecular weight is very less. Then only it can be easily transferred to the host. The process can be easily carried out. The nature of this is very small and the molecular weight is very low. Then this vectors, another property of vector is, this vectors contains a O or I region, that is origin of replication. There is an origin in this plasmid, there is an origin of replication. It initiates the replication, origin of replication, and there is a marker. There is a selective marker is present to identify the transformer. So what is the gene transferred? What are the sequences or transferred? It can identify. So it permits, it allows, selectively it permits the invaders, foreign invaders. So marker, and there is a cleavage site. So these properties are present in the vector. So these three are very important features of the vector. Look at this diagram. See there, this is the plasmid. So in this plasmid or vector used as a plasmid, or it is called as cloning vehicle. So in this there are three important regions are there. This region is called as ORI, origin of replication. This is the origin of replication. What's the role of this? A cloning vector must contain an origin of replication which recognized in the host cell. So origin of replication recognized the host cell so that it replicate along with DNA. So along with DNA, the DNA that it carries, it can replicate. So this ORI or origin of replication is the main feature of this plasmid or vector. Second one, selector marker, this region. This region is a selectable marker. So, what is the role of this selectable marker? It enables cell containing vector to be selected and identified. The altered gene can be identified by this markers, selectable markers. And there are number of cleavage sites. Here these all are the cleavage sites. What are the cleavage sites? PSD1, BAMH1, SAL1, ECO or 1. So, these enzymes, this cleavage sites are use more enzymes. So, in this site it used one or more enzymes can be used. Either one enzyme or more enzymes can be used for this cleavage site to cleave this plasmid. So, this is the vector which contains 3 recognition regions, 3 important features. This is the 3 important features. What are they? Origin of replication, then selectable marker and cleavage sites. These 3 are very important in vectors. So, once again recall what you learned today. First question, today's topic is tools for genetic engineering. What are the tools? Enzymes, vectors and host organism. So, today you learned only 2 topics, enzymes and vectors. So, next class there are different types of vectors. You are going to learn different types of vectors. So, today you learned this question, restriction endonucleases, how it acts as a molecular scissors. Then this tablet column, there is a tablet column given in your book. Learn the type of restriction enzyme, what is the microbial source, what is the recognition sequence and what are the fragments, how it cuts. It cuts in the blunt end or in the sticky ends. Then finally you learn the vectors. What are vectors? First question under vector is what are vectors or define vectors. Then what are the properties of vectors and what are the features of vectors. That is the thing you are going to learn today. Prepare well, daily portion you will learn thorough. Thank you.